Hi everyone, and this is Naidu. I'm from Great Online Training, and today we are going to learn about clinical SAS and what exactly the relation between clinical SAS and our general SAS software. And apart from that, what are all those things included in clinical SAS and why we are calling it as a clinical SAS and all those stuff we will see. So introduction to the clinical SAS. So what is introduction to clinical SAS? So generally we have a SAS software, and why they are using a SAS software in a clinical sector. So now you just go here and see this. What is the relation between SAS and clinical related items in the sense of clinical trials? So most of you people already know that so most of the drugs being sales are being so what is it consumed by the persons are also patients. Those drugs all are from FDA approved drugs. If any pharmaceutical company or else any pharmaceutical what is it organization, they want to release the drugs into the market, compulsory the drugs need to be tested. So tested in the sense how they can say it is tested, they need to do a clinical trial on the particular drug. Based on the clinical trial, that reports will FDA will looking into that. If all the reports are perfect in the sense so each and every drug having some side effects, but the side effects should be very lesser than our benefits. So then only the drugs will be get approved by the FDA. So once that particular reports are reviewed by the FDA people, okay, this drug they can release into the market. Then that particular drugs will be released, and then that drugs will be selling into all the so countries and all. But which reports they are going to looking into that? So FDA guys, which reports they look? The reports which are generated by the SAS programming, that reports they will just have a look and they will just have a review on it, and then they'll release that particular drugs into the market. So generally, so when I say it is a clinical trial, so what is the overall picture of the clinical trial? So now you can able to see that so generally what clinical SAS programmer will do. So clinical SAS programmer will read the protocol. So based on the protocol, he'll try to understand so what we need to do as a SAS programmer. Protocol is a document. It is just our PDF document only, but it have all the information about your clinical trial. So nothing but so what the physician need to do. That means we call it as in clinical programming principal investigator. What principal investigator need to do, what statistician role is, what the programmer role is, how many patients is required, and how many patients is need to enroll from which country. So like that all the information will be there in the protocol. So it is a approved protocol from the FDA. So once they have done with a preclinical trials, that means animal testing whenever they have done, then they will give a permissions to do the clinical trials. When I say clinical trials, it's a compulsory on human beings. So once the clinical trials is done and the protocol is ready, and they'll send this protocol to the FDA for approval purpose, that means this is all we are going to do in this particular clinical trials, then they will give an approval for this particular protocol. So based on the protocol onwards, next step is for we can say for the CRO companies, what they do is they'll look for the investigators and hospitals. Imagine they want to release the drug in the China. So compulsory they need to do some clinical trials in China. So that what they do, they look for some hospital who can provide the patients who are suffering with our diseased means our targeted disease. Imagine we have developed some drug called X. We means here CRO companies. So the CRO companies developed some drug, they want to test the drugs. Compulsory for testing purpose we need to have patients, and as well as we need to have doctors. So in clinical terminology we call it as a instead of doctors, we call it as a principal investigators, or else we can call it as a PI. And apart from that, instead of patients, we call it as a subjects. So patients we call it as a subjects, so that we need to go for looking for a subject, and as well as investigator selection also. They'll target for different hospitals, they will target it, and they'll take the investigators from there. Investigators in the sense, the person who is responsible the activities whatever happen in that particular hospital during this total clinical trial period. So that particular person we call it as an investigator for that particular hospital, so that he is the responsible for all those activities, what to done and what not to done in that particular hospital to these patients. So that is your investigator selection. So once the investigator, so the CRO company's investigator selection process is done, then each and every or every country wise they have their local ethics committees. We can call it as a local boards, we can call it as a. So that means if they want to do a clinical trials, so initially they want to apply for permissions to do the clinical trials. FDA has given a permissions to release the to do the clinical trial, but we need to get the approvals from the local ethics committees also. So once they have given a approval, okay, they have, okay, you can do the clinical trial, that means CRO companies will do the clinical trial, then they start recruiting the patient. So who are all the patients here? The patients in the sense, the people who are suffering with our disease, and apart from that, in protocol we can say a set of rules. So set of rules in the sense, the patient must be 18 years above old, and patient should not be alcoholic, patient should not having a smoking habit, patient must must complete the previous dose of the 300 mg dose or something like that. So like that, there is a set of rules, we can call it as a, those we can call it as a inclusion and exclusion criteria. So that the inclusion and exclusion criteria, they will looking into that, and based on that they will recruit it. If you just see, in around 7 patients we have only 1 patient got selected. The reason behind that is, the patient fall under this category, it have all the all the criteria he met. He is 18 years old, he don't have a smoking and habit, and he don't have alcohol habit also. So that's the reason why that patient is participated into the clinical trial. Imagine if we need 400 members as a patients, compulsory they will screen around 600 or 650 members, they will screen it. So most of the cases, around 20% is the screen failure rate. Screen failure in the sense, the test what we are doing is screening. Screening means we will check all those activities, what is required for us, is it there or not. If those activities means those diseases or else those habits and all, so if it is there, then he'll follow the into a clinical trial, otherwise he will be excluded from the study. So those patients we call it as a excluded patients. So this patient we call it as a included patients. Once the patients are included, then we start taking the information from CRF. So we call it as a in real time case reportive form. So the patient came to the hospital, but we are taking an information like age, sex, height, weight, race, ethnicity, and all those things we are taking it. But where this information will store? Will store it on a white paper? No. CRF, nothing but case reportive form. The case reportive form is a document, we use it as a to taking the information from the patient. Case reportive form in the sense, it is exactly like our biodata form. So whatever we have it in biodata form, we will have the same fields. Biodata form, actually what we have, like a name, address, email ID, phone number, and educational qualifications and experience, we will have it in our biodata. But patient information, like in CRF, patient ID, patient race, ethnicity, sex, gender, and all we can able to see in one CRF, that is called demographic CRF. We have a different CRFs. We are going to talk about in detailed manner later on, and first let me talk about a case reporting form. So case reporting form is the tool to just to pick all the information from the patient. Nothing but age, okay, 28, they will enter. Case report form in the form of paper only for nowadays. So what they do is, the doctor beside there is some person who is caring of all those information from the patient. Okay, tell me your age, tell me your sex and ethnicity, race, gender, and all those stuff. Whatever is required, they will take it from the CRF. That CRF is nothing but information about your patients. So one patient for one CRF for one visit. Like that, multiple patients, multiple CRFs, and multiple visits information will captured, and that CRF will send it to the data entry and validation team. So here you can see there is a lot of people who are just entering the data into the databases. So whatever the data they have collected it from there, they'll enter the data into the database, and then so once the data is entered into the database, so this is how they are doing the database. So here we can see DB load and DB lock, and before going to that, there is a data entry and validation, I said. So what is data entry and validation? What exactly it could be done? Nothing but imagine, so whenever the person is entering the data, imagine I'm the person entering the data, some X data I'm entering it. X data in the sense the list of people information, the list of patients information I'm just entering into the that particular database. The same work will give it to you also to do the same. So what happened? You all also enter the same data into the database, and you also enter the same data into the database. So what happened? What is the use? Based on that, there is a chance I do mistake somewhere, but there is no case like you compulsory need to do the same mistake at the same place. Maybe you will also do the mistake, but in a different place, or else a different patient means so data you entered, you may do the mistake. So in that case, what happened when they are checking the data between my data with your data, where exactly the wrong is between, so they'll compare it, which one is the right and which one is the wrong, they will decide it finally, and then they will conclude it. So this we call it as a double entering of the data, and apart from the double entering of the data, so this particular all the process, our final motto or final aim is to avoid error free data. So that means to reduce the discrepancy in the data. We call it as a terminology in clinical sector called discrepancy. So they'll try to reduce the data discrepancy. So once the data discrepancy is reduced, and they'll enter the data into the database, they'll keep on enter the data into database. Imagine around 5 people. Imagine the clinical trial is 400 people, they recruited 400 people. First time the patient came to the hospital around 10 times. First time they came, they take the information, that information they will send it to the CRF and all to the this particular data entry and CDM team, clinical management data management team. The clinical data management team will enter the data at a first visit information. When again the patients is came for the second time, so that again second patient time will come the data and they'll enter it again. Third time, fourth time. As per the protocol, how many times we said to the subjects to attend the clinical trials will be based on that. Those many times the patient or the subjects will come into the clinical trials, and then keep on taking that information in CRF. That CRF will send it to the CDM team, clinical data management team, and clinical data management team will keep on the data into the databases. But here you can see there is a two things called DB load and DB lock. So what is DB load and what is DB lock? So DB load in the sense they'll keep on enter the database into database, that is called loading the data. But there is another term called database lock. At certain point of time, so there is no incoming flow of the data. That means investigators have a green signal that so we have completed all the visits what we have in the protocol. Based on the protocol, we have completed all the study. There is no incoming flow of the data into the database. You can lock it. So CDM people will just looking into that, all those things is ready, so that they will take a just information from us, like whatever the data we got it or not, is there any still data discrepancy, or else is there anything which is not captured, or which is that. See, if we have something, an issue with the data, we are not the responsible persons for that. We need to just flag it, there is a data issue somewhere. Okay, that data issues will be taken care by the CDM team itself. We are the programmers here. What we do is, whatever the data is available in the database, we try to use the data and creating a reports as per the FDA submission. That's what our job is. So what happened? So they will ask us, okay, is there anything like, it is just normal check only, it is not mandatory compulsory. You need to, means as a programmer, SAS programmer, you no need to give the message, means information like yeah, all the data is clear, I have checked everything, and something like that. It is not required. We just tell them, okay, what we got it up to this, it is fine, and but so they will check in their end all the data is got it or not. So based on that, so the database lock. So generally what is the problem and why they are checking with us means, see, if we are doing a clinical trial process of the report generation, we have some issues, but those issues will not caught by CDM people, but still we are not informed those issues to the to the team of the CDM team. So then those are outstanding issues. That means if they have not changed and then they have locked the database, so that locking a database in the sense they are not putting any lock to the database or something like that. That means there nobody is going to work on it. It is going to be freeze. That means whatever the available data we have it, we need to use the same. The reason behind that is database lock is, FDA is very important in database lock and all. So whatever the reports we got it, based on that results we'll try to produce some reports. But there is always chance that if we results are not perfect, then there is a chance we can unlock it and then we can do some changes or something like that. To avoid those things, FDA is very concerned about, if the database unlock happen, there is a crucial part in a clinical sector. That should not happen anytime. The database lock should not be unlocked at all. Once they freezed it, that's it, that is the final thing. So then the report, SAS people will generate these reports. Those reports in the form of tables, listings, figures, we call it as a data sets, tables, listings and figures. Those reports we will send it to the result meeting. Result meeting we can call it as a like the people who are spending money, that means mostly the investors, and as well as trial statisticians, trial programmers, all will involved into that particular result meeting. Trial statistician in the sense the statistician who are working in this particular clinical study and who are all responsible for all the statistical things what happening in this particular total data analysis. So that person is your trial statistician. Trial programmer in the sense the programmer who is involved to creating reports and all from starting to end. So mostly trial programmer will involve only those, because you will not get invite, even though if you get a job as a SAS programmer, you will not get the invitation to participate in the result meeting. Understood? Result meeting in the sense which is very secrecy one. So that means how the results are gone. Based on that, there is a fluctuation in stock market also. So there is a lot of people expectation on that. Yes, there is upcoming drug in coming into this particular Novartis drug is coming, so that drug will change the future of Novartis company like anything. But the result meeting, they come to know that the results are not that much good. So then it will affect our stock market and all. Mostly those reports and those things will not be will not be disclosed at anywhere. And now the result meeting, when they satisfied yes the results are all getting perfectly, then they will submit these particular reports to the FDA. So FDA will approve these particular drugs, so that this particular report, then only our clinical drug, whatever we have done the clinical trial. So mostly the clinical trial not only for drugs, mostly for syrups and some injections, maybe some what we can say, all the medical devices. Suppose imagine they have invented some medical device that is a pacemaker. So pacemaker in the sense it is instead of heart it will work like that, it is working like ours. So even the heart also they want to release into the market, compulsory need to do the clinical trials on that also. So these are all are mandatory. So the clinical trials SAS programmer do exactly talking about data analysis and reporting using SAS software. Clear? So this is the overall picture of the clinical trial programmer role, what need to be done. And next, so introduction of SAS program. So most of you people already aware of it. So whatever we have, the raw data, that means the data which is available in data, that means that particular database, that we call it as a raw data. The database data we will call it into SAS software in the some form of a data step. So once we create the data step, and we will create a SAS data sets. SAS data set in the sense it is a collection of tables and rows. Will create as a SAS data sets. Those data sets will use it for a reporting purpose. So those reports will be generated. This is the workflow of SAS programmer. Wherever you go in any of organization, they will give you the raw data. You need to produce the reports. This is the starting point and this is the ending point of your programmer level. So generally my question is, why pharmaceutical companies depends on SAS programmer, what they need? So why the only SAS programmers in a clinical sector is required? That means we need to produce certain reports. Those we call it as a tables, we call it as a listings, we call it as a graphs of figures. This we call it as a TLFs, or else we can call it as a TLGs. So the person must have an experience on creating a tables, listings, graphs and figures. Then only the person can survive in clinical sector job, clinical sector SAS job mainly. And apart from that, we need to create a data sets also. So those data sets I'm going to talk about in later slide. So this compulsory need to have a good idea, each and every person who are participating in a clinical SAS programming role. Compulsory they need to aware how to create the table, they need to aware how to create the listing, how to create the graph and as well as figure. And next thing is like examples of a table. So this is how the table is looks like. You just see the table, demographic and baseline characteristics. In that age wise, how many people we have it, around total number of people in that particular clinical study is around total number of people in clinical sector for that particular treatment group wise 31 and 29, total 60 members are participated in the clinical trial. Based on that, females are 22 for this particular active drug taking people and 9 members active drug taking people. And here you can see that it's in a placebo, 29 members in that 29. So out of 16 is male and 12 is a female or something like that. Maybe some values are missing or something like that happen. Because of that reason, there is a chance to having a count is less than our n count. The total number of people who participated in the clinical trial is 29, but here you just see 16 plus 12, 18 only. One patient got missed here. Maybe there is a chance to that particular person has gender is missing. In the sense, due to some reason, maybe that is not captured, or maybe the data records are not there in our database, or something happened. Because of that, the count is bit less than our total count, right? So now, so this is all the information. So how the table is looks like. You can see how the listing is looks like. The difference between table and listing, table is nothing but so top to bottom, it is nothing but a summary of output. Summary in the sense, imagine if it is around 500 members participate in the clinical trial. So the 500 members data will be just like males are 200, females are 300 or something like that. That means summary of your outputs, we call it a summary of your data, we call it as a table. But listing is nothing but the total 500 people information also we will display it. That is the best difference between table and listing. And there is one more thing called a figure. So mostly figures, what they do is, it is just for to see the data, whatever the data we produced in the form of tables, the same data they need it as a figure sometimes. That means they want to see the data in a pictorial representation so that it will be a bit easy for them for publications, for them to display some information. Yes, our trend is going like this, our drug is working fine. So instead of having a values in a table, so just making as a some graph, and so that how the graph trend is going on, it's a keep on increasing or else keep on decreasing and all those stuff. So it's a easily understandable to the person so who in FDA or else maybe for any submission process. So that's the reason why figures also important. And this is the latest one in the sense, so compulsory the clinical SAS programmer, the person who want to do a clinical trial process, a clinical trial SAS programming job, he need to have an experience on CDISC, that means Clinical Data Interchange Standard Consortium, SDTM, so nothing but Study Data Tabulation Model, and ADM. So what happened? Apart from TLFs, tables, listings and figures, compulsory the person need to have an experience on CDISC, like Clinical Data Interchange Standard Consortium, and as well as DDM and ADM. So what is this CDISC? Is nothing but CDISC is a organization, so it's a free organization only. So the people will give a set of rules. They already kept a set of rules to do the standardization in the sense, see, I'm a Redis like a company, and another person is another company is Pfizer. They are doing clinical trials, they are sending a data to the FDA, and I'm also doing a clinical trial, I'm sending a data to the FDA. But the problem is, every person every organization, they are following their own standards. But CDISC people, what they have done, they generated two things. One is SDTM and ADM. So SDTM in the sense, whatever the study data in the sense, the data which is collected directly from the patient, that we call it as a source data. That source data always in the form of SDTM, that is Study Data Tabulation Model format. Let it be any organization, all the organization need to follow the same rules. The rule in the sense, suppose imagine we call it as a subject ID. Another patient another organization, they are following it as a patient ID. But it should not be the different names, it should be the same name, like patient ID and subject ID will be the same. Okay, that means it should be subject ID. He also need to follow as a subject ID, I'm also need to follow the subject ID. So this is SDTM is nothing but to submit a raw data to the FDA. ADAMS is nothing but analysis data models. Analysis data models in the sense, if they want to send the data in the form of analysis data sets, nothing but, see, there is a chance that whatever the data we have captured it from the patients, we will not use all the data for our analysis purpose. What we do is, we try to create few variables and few observations containing a data as a special data sets, that we can call it as a analysis data sets. That means the data sets which is used specially for analysis, those data sets we call it as a ADAMS. We need to follow the ADAMS standards also. That means whenever the data you are submitting to the FDA, ADAM data sets also need to be standard, so that SDTM and ADAM standards in every organization nowadays they are following. So compulsory you need to have an idea, so what is SDTM and what is ADAM and all, and then only you will be a perfect clinical SAS programmer job, you will fit it. But there is a chance it is a two things, like so jobs are like mainly for TLFs, tables, listings and figures, or jobs are mainly like CDISC, nothing but SDTM ADM, people who already know the SDTM ADM also. So that you decide. If you know everything, then you have a job of scope is more than normal people who have only know the TLF. Okay, that you decide. And this is the CDISC. The CDISC people, SDTM and ADAMS will create it, who will send that SDTM ADAMS to the FDA. Right. So the next one is, you just see this, these are the list of companies what is available in US, like how they are sending a data. Even GSK, Roche, Merck, and Novartis, Pfizer, Abbott and Lilly. So these are all the pharmaceutical companies. Whenever they are submitting a data to the FDA, it should be in a SDTM standard format. Then only the FDA will approve this particular drug. So nowadays, so step-by-step clinical trial programmer role. Earlier what I said like a overall picture of the clinical trial I said. In that I have said only one piece of information, what is clinical SAS programmer role. But now it is in detailed manner of clinical SAS programmer. Whenever you get a job as a SAS programmer, a clinical SAS programmer, first of all you need to have an idea on trial protocol, and then you need to have an idea on CRF, then you need to idea on eCRF, electronic format of CRF. The difference between CRF and eCRF is, CRF in the sense it is just a paper format of output. eCRF is nothing but a electronic format of output. We can see it as a PDF document. eCRF. So once the eCRF, we need to understand what is the annotation. So in the sense, they said ID, patient ID, but the patient ID, how it is captured as a subject ID in our data set, means annotation is nothing but it is clearly saying that which field on the CRF is converted as a variable. Imagine it is in a sex. Sex is the field, we have it in CRF, how it is converted as a variable. So that in the sex place itself they will clearly write a annotation that, so annotation in the sense there is writing a label. Okay, sex is nothing but sex is the variable name. Suppose it is a, imagine it is in a date of birth, DOB is the variable name. So that is called annotation. The CRFs after annotation will be called as a annotated CRF. So that annotated CRFs will submit for SDTM creation. The people who are working on SDTM datasets, they'll try to use the annotated datasets, and they'll try to use another document called SDTM IZ, SDTM implementation guide. That guide also I'm going to explain these things later on. First you need to understand SDTM, and based on the SDTM they will create some datasets, all the SDTM datasets whatever is required. So whatever all the documents, whatever all the datasets we need to create and we need to submit for FDA, that will be already decided and that will be already there in SDTM implementation guide. When we are talking about SDTM in detailed manner, then we will see what is SDTM and what all the information we'll have in SDTM, and finally they are submitting these reports to the FDA. This is one branch in the sense the people who only work with SDTM datasets to creating SDTM. There is a chance to people work on SDTM to ADM conversions also. So that what happened, the SDTM datasets they'll take it from SDTM to ADM. ADM in the sense, just imagine, I will give an example like yes, so they have collected some information from the patient. The patient came to the hospital around 10 times, but he came 9 times. One time data is not there. So SDTM dataset, we are not changing anything like that. It will be like 9 times he came to the hospital, so that 9 time information only we will have it. But ADAMS will try to impute that record. We will take it that record from the previous data results, whatever we have it, that we will take it from there and we will try to create it in ADAMS. Like that many things we will do, all the derivation parts and everything. They will give you, wherever you get a job as a SAS programmer, they will give you ADAMS specs. So analysis data model spec, so based on that you need to derive analysis data sets, we can call it as a. So once this analysis data sets are ready, so that analysis data sets we will use it for creating a tables, listings and figures. So once the TLFs are ready, again the TLFs also we will submit for FDA, and this ADM data sets also will submit for FDA submission process. So this is all the overall clinical trial process in mainly for SAS programmer role in detailed manner. Except this, there is nothing into your clinical domain. This is all those things you need to know as a clinical SAS programmer. And next one is here. That means this course anyone can learn, like the postgraduates, graduates, BTech, MCA, MSc, and all the pharmacy people who anyone want to learn, they can learn this course. And so what is the job scope at your place means most of the people they are already aware of it. There is a good job scope in market right now for a clinical SAS programmer job, and apart from that it is a work from home jobs are more in clinical sector. So that's it. So this is all about the overall view of a clinical trials and what I have expected to deliver today. So like so I just given all the brief information like what is clinical trials and what is the process and all those stuff. So this is all about a clinical trial process. Thank you for watching. Bye.